Good morning. I'd like to begin by thanking the organizers for this opportunity to speak with you today about our novel MOA reflective bioassay for quantifying potency of therapeutics targeting the SIRP-alpha CD47 signaling axis. Before I begin, I'd like to introduce you to Eurofins Discoverex for those of you not familiar with us. So Eurofins Discoverex is a product-focused company that's based in Northern California, and we have developed over the years over 800 off-the-shelf assays for in-house drug development. Our primary expertise is in cell line engineering and cell-based assay development, and we have significant expertise in bioassay development, optimization, and qualification for late-stage drugs. Importantly, our technology has been successfully implemented at global pharma, biotechs, and CROs, and they've been implemented across the drug discovery continuum in early discovery and in development. And as important is that these particular assay technologies have been implemented in lot release for several marketed biologics in the US and EU. Our portfolio is quite broad, and it supports discovery through lot release for a variety of target therapeutic areas and different targets such as GPCRs, interleukins, and more recently immune checkpoint modulators. And so today's discussion, of course, will be focused on one of these for SIRP-alpha. So assays that are used in discovery and through lot release, they have different requirements, but they all must be fit for purpose. And of course, for those that are used in late stage testing or for QC lot release, they can be used also for characterization or process development, but they're primarily focused on relative potency, which is evaluating a test sample relative to a reference standard. And because these are analytical assays, they have very specific requirements that must be met and are expected to be met by the regulatory authorities. And among these are that the assay is reflective of the MOA of your drug, they're robust, accurate, have adequate intermediate precision, and demonstrate linearity over a specified range of concentrations. And importantly, they need to be stability indicating, or show the ability to detect changes in your drug due to degradation or sample storage. And also, they should be amenable to transfer to multiple sites when you're doing testing in multiple CROs globally. For these reasons, implementation of cell-based assays for lot release typically focuses on the use of engineered cell models rather than using primary cells, and where possible, assay-ready cells rather than continuous culture for the ease of setting up assays. And finally, robust and easy to transfer protocols are a must. So if we talk about the SIRP-alpha CD47 axis, I think we all agree that it represents an innate immune checkpoint that is growing in great interest. And this particular conference is focused on the biology here, so I don't need to really reiterate it. But essentially what we're trying to do is identify therapeutics that will perturb this SIRP-alpha CD47 axis to reinitiate the phagocytosis of tumor cells. And not only does blockade of this axis promote phagocytosis, but it's also thought to enhance adaptive immunity by priming anti-tumor cytotoxic T cell responses. So when we decided to build an assay to support therapeutics, quantitation of therapeutics targeting the SIRP-alpha CD47 axis, our goal was to move away from using primary macrophages for the reasons that I described earlier. So what we chose to do is to look at the molecular mechanism of of this signaling cascade. And what we see from this cartoon is that the extracellular domain of SIRP-alpha engages its ligand on the surface of another cell, typically the tumor cell. And this results in an intracellular cascade of phosphorylation, which results in recruitment of SHP phosphatases that ultimately result in inhibition of phagocytosis. So our goal was to build an assay that would capture this early event that was reflective of receptor engagement and ultimately resulting in the MOA that is desired, and that is building an assay to monitor SHP recruitment. So we did so by using our internal technology, which we call enzyme fragment complementation, and that technology is shown here in a cartoon on the left. So we use our the EFC technology is based on a fragmented beta-galactosidase enzyme, where we have two inactive fragments shown by these two cartoon characters. And when the two fragments are separated, there is no intrinsic enzyme activity, but when they're brought into close proximity, we're able to reconstitute an active enzyme, and we can detect that by the addition of substrate and detection reagent, and this results in a chemiluminescent signal that can be read on an appropriate plate reader. So in designing our assay, we developed this as a co-culture model, where in the Jurkat cells here, we're showing expression of a full-length SIRP-alpha, and at the C-terminus, we fuse our small, we call this an enzyme donor tag, and in the same cells, we co-express the SHP phosphatase fused to the enzyme acceptor, so the other half of the beta-gal reporter. So in the absence of ligand, these two proteins remain separated, but when you co-culture these cells with ligand-presenting cells, in this case another Jurkat cell expressing CD47, we're able to result in the two proteins coming together, reconstituting the active enzyme, and we can detect that by the addition of our substrate. Similarly, if we block this interaction with agents that target either CD47 or SIRP-alpha, we're able to inhibit that dose-dependent in a dose-dependent manner, and that results in a loss of signal. So we've demonstrated this with a number of different agonists. What we found to be most efficacious is a cell-presented ligand, as I described in the previous cartoon. And what you can see is that a typical dose curve of CD47 presenting cells gives you a dose-dependent increase in signal. And typically what we're looking at is somewhere around a 20-fold window. And similarly, if we block that interaction with an anti-CD47 antibody, as shown here, we get a nice dose-dependent decrease in signal. And again, an assay window around, in this case, around 30-fold. And the IC50 is typically, depends on your antibody, but around 50 to 100 nanograms per mil. So we've established a stable cell line that stably expresses SIRP-alpha. This expression is stable over 45 passages, and we've determined that it varies by less than 20% over those 45 passages. And importantly, the functional response also varies by less than 20%, both respect to the actual signal to background and also with IC50 for the antibody. So a very robust cell model. So one of the questions that we often get is, how do we know what we're measuring is actually SHP recruitment? So what we've done is actually developed a number of mutants by mutating these ITIM motifs in the C-terminal tail of SIRP-alpha. These are the four tyrosine residues that are thought to be phosphorylated and are important in the SHP phosphatase recruitment. And for the sake of simplicity, I'm showing just a single mutant here where we've mutated all four of these residues to phenylalanine. And what you can see is that mutating these four residues results in the inability of this SIRP alpha mutant to signal at all. So it fails to respond to CD47 presenting cells, whereas the wild type SIRP alpha gives you a nice robust response. Similarly, if we look at the inhibition mode, because we're unable to stimulate these cells, we see no response to an anti-CD47 blocking antibody, but you get a nice dose-dependent response with the wild type. So what we've seen is that single mutations were successful in disrupting signaling to different degrees, some not very strongly, but mutation of 3 or more of the tyrosine residues completely abrogated CD47-mediated SHIP recruitment, which indicates that this is the mechanism that we're actually detecting with our assay. So we've evaluated the reproducibility of the SIRP alpha signaling assay in a number of different ways. This is just an example of data from a single analyst who's run the assay on 3 independent days. And it gives you a good idea of how robust the signal is and reproducible in terms of IC50 and signal to background. And even hill slope is quite similar between these different experiments. And similarly, if we look at the data with respect to relative potency, so comparing 2 different curves of the same concentration, each of them at 100%, we can see that on 2 independent plates, we're able to see very similar relative potencies, and the actual relative bias is quite low. And the average relative potency over these 5 experiments was 99.5% for a 100% sample. So quite good accuracy and very good intermediate precision. So of course, it's much more straightforward to transfer a cell line, especially, an assay when it has been converted to an assay-ready format or frozen ready-to-use format, especially when you're considering that this is a co-culture where you would normally have to culture two different cell lines independently and keep those ready to run in an assay. So we have converted this to our frozen ready-to-use method and changed the protocol slightly to simplify it. But essentially what we do is we have frozen a vial each of CD47 presenting cells or our SIRP alpha cells, and we thaw a vial of each of those, add them to the assay plate, prepare the antibody dilutions and add those to the assay plate, and everything is incubated for 24 hours prior to addition of the detection reagent for 1 hour, and then we read that on our plate reader. So the total assay time is roughly 26 hours, so easily handled in most CROs, very easy workflow. And what you can see is that the data look very similar to what we see in continuous culture, which is shown here on the left, and the cryopreserved or ready-to-use format on the right. And again, hill slopes are very similar, IC50s very similar, and also a comparable signal to background. So we moved forward with this assay format to establish that it's fit for purpose for potency and lot release by performing a qualification study. And this is performed per ICH guidelines to establish accuracy, precision, linearity, and stability indicating properties of this assay. So we did this using 2 analysts over multiple days and evaluated 5 different concentrations over a range of 50 to 150%. And each concentration was evaluated 3 times by each analyst over a minimum of 3 days. So each concentration we had 6 values. And what we used is each sample was evaluated in duplicate in this interleave format that is shown here on the right in an 11-point dose response. And in the same study, we also evaluated specificity and force degradation samples. And this table here summarizes the results from that study. What you can see is a lot of numbers, and they're very small, but ultimately what we were seeing is really excellent accuracy, close to 100% for all of the values that were tested, very low relative bias, and an intermediate precision of 6.5%. And dilutional linearity, R squared, was perfect. So it was really, really performed very well. And I can say that this particular qualification was performed with just a commercial grade anti-CD47 antibody, but we've seen similar performance with other types of molecules as well. So in addition to the excellent accuracy and intermediate precision, what we see is that the assay is highly specific. If we run antibodies targeting other checkpoint receptor ligands, such as PVR, CD80, or PDL1, we see no activity in the assay or no significant activity, whereas the anti-CD47 antibody gives a nice, robust dose-dependent response. Similarly, we've tested a number of different IgG, human IgG molecules, and they are essentially inactive and very much less potent than the anti-CD47 antibody we used here. And probably most importantly, if we perform forced degradation of our antibody at 65 degrees for 17 hours, we are able to detect a significant difference, right shift in the sample, suggesting that the assay should be stability indicating as well. So the data that I've shared with you thus far has really been focused on SIRP alpha variant 1, which is one of the two most common variants, most prevalent in the population. And as I know this audience is aware, SIRP alpha variants 1 and 2, which are the two most prevalent, differ by only 15 amino acids, as is shown in this, this cartoon here from the Hatherley paper. And what they observed by several techniques is that X-ray crystallography being one, is that the contact points between CD47 and SIRP alpha are actually pretty well fall in pretty conserved regions between the two variants. And they also determined that the KDs for binding to CD47 are not too different between the two SIRP alpha variants. However, for clients who are targeting SIRP alpha variant 2 or generating molecules that are SIRP alpha targeted, there may be some relevance to having both variants or one or the other. So we have developed an assay for SIRP alpha variant 2. Shown here up on the right is a comparison by flow of the expression levels of SIRP alpha in the variant 1 versus the variant 2 assay. You can see that they're quite comparable. And what we've also observed is that the response to at least an anti-CD47 antibody in the two assays is also very similar, both with respect to IC50 and signal to background. So this assay is also available for testing. So I'll just close by sort of reiterating that we have generated an engineered cell system that measures activation and inhibition of the CD47 SIRP alpha signaling axis that does not use primary macrophages. It is a mechanistically relevant endpoint looking at SHP1 recruitment, and it robustly quantifies CD47-mediated SIRP alpha signaling. As I mentioned, we have assays available for both variant 1 and variant 2 of SIRP alpha, and these assays are suitable for not only anti-CD47, but SIRP alpha targeted therapeutics as well. The assay delivers a highly specific response to stimulation with CD47, and it is, we've demonstrated, at least with the variant 1, that expression of SIRP alpha and the functional performance are stable over at least 45 passages. We see excellent intermediate precision, accuracy, and dilutional linearity in the bioassay, and believe we have demonstrated that this assay is fit for purpose for potency and stability indications. I didn't show any of this data, but the assay is also tolerant to human matrix, so it may be suitable for neutralizing antibody detection applications as well. And it is available not only in continuous culture, but in the ready-to-use assay format that I shared with you. So I'd like to close by giving you information about how you can reach out to us should you be interested in this assay or the development of other assays that are similar to this one. And I'll thank you for your attention.